Signal transduction. Hormones are responsible for several actions that take place in the human body, and they carry out these actions by communicating with cells and cell tissue, ultimately to maintain homeostasis. Example, when blood sugar is low in the body, adrenal glands secrete epinephrine to tell the liver to increase the breakdown of glycogen to create glucose. The way hormones and cells communicate with each other is very distinct because, unlike humans, they cannot simply talk to each other. Instead, hormones bind to specific receptors in the plasma membrane to pass on a signal. Signal transduction specifically relates to hormones that cannot pass through the plasma membrane. During signal transduction, hormones or ligands bind to a metabotropic membrane receptor. This is basically a receptor that acts with the help of a G protein. The ligand binds to a receptor. The receptor undergoes a conformational change. This basically means that the receptor changes its shape. When this happens, the membrane receptor grabs the attention of a heterotrimeric G protein. The name heterotrimeric comes from the fact that the G protein is comprised of three parts: an alpha, beta, and gamma subunit. In a rested phase, the alpha subunit has a GDP molecule attached to it. When activated, GDP converts to GTP. This conversion will always take place in the alpha subunit. When GTP is bound to the alpha subunit, it dissociates itself or splits apart from the beta and gamma subunit. It was once thought that beta and gamma subunits did little in the process of signal transduction. However, research shows that the beta and gamma equally contribute to the activation of target proteins. Specifically, however, the alpha subunit will find and activate an enzyme known as Adenylyl cyclase, which will convert ATP into a molecule known as Cyclic AMP. In this process, ATP will take away two phosphates, creating adenine monophosphate, or AMP. As for where the word cyclic comes from, it has to do with the shape of the molecule. Cyclic AMP is important because it acts as a secondary messenger that will finish carrying out the original message throughout the cell. Cyclic AMP will activate kinases. In the case of epinephrine, it will activate protein kinase A. Protein kinases, much like A, are basically enzymes that attach phosphates to other molecules. This process is called phosphorylation because phosphates are being added. The process of phosphorylation will in turn cause a cascade of reactions that will eventually cause a cellular response in target cells. And that's basically how it works. A single ligand will bind to a receptor, which will then activate a G protein. The G protein will activate adenylyl cyclase, an enzyme that converts ATP into CAMP by taking away two phosphates. Cyclic AMP will then activate kinases, proteins that will cause a cascade of reactions that will activate other enzymes and accelerate metabolic activity of a cell. As a result, a cellular response will occur, such as the increase of glycogen levels in the body. When an action has successfully been carried out by target cells, an enzyme known as Phosphodiesterase inactivates cyclic AMP. This is important because without phosphodiesterase, levels of glycogen in the bloodstream would be too high.